Welcome back for the last session of the meeting. We're going to be talking about emerging SARS-CoV variants. And Dr. Scooby, please go forth.
Good afternoon. Next slide. Next slide. There are multiple SARS-CoV-2 variants circulating globally. After emerging, some disappear and others persist. CDC and others are studying these variants to understand whether they spread more easily, cause milder or more severe disease, are detected by available diagnostic tests, respond to current therapeutics, or change the effectiveness of COVID-19 vaccines. Based on the evidence, some variants are classified as variants of concern and appropriate public health actions are taken. Next slide. Viruses constantly change through mutation, so new variants are to be expected. SARS-CoV-2 has a low mutation rate compared with influenza and HIV. The drivers of evolutionary selection for SARS-CoV-2 are still being characterized, but may include chronic infection, interspecies transmission, therapeutic treatment with antibodies, prior immunity to strains with limited cross-reactivity, increased transmissibility, or the founder effect, where a small number of genotypes seed a new population by chance. Next slide. As an example of SARS-CoV-2 strain replacement is the D614G variant. As shown in the graph, this variant was discovered in January of last year and increased to almost 100% prevalence worldwide by June. Compared with the ancestral strain, this variant has increased infectivity, which is attributed to a more open conformation of the viral spike protein that helps it bind to host cells. Next slide. The spike protein is shown in red at the upper right, studying the viral surface. The D614G variant likely has increased transmissibility, giving it selective advantage, but it's not more clinically severe and current vaccines are still highly effective. So this variant isn't of particular concern, but it highlights the potential for similar replacements to occur with variants that may be more problematic. Next slide. There is general consensus that 3 SARS-CoV-2 variants are variants of concern. B117, first detected in the UK, B1351, first detected in South Africa, and P1, first detected in Brazil and Japan. By now, these variants are being detected in many countries. Both B117 and B1351 are estimated to be 50% more transmissible and have become the dominant strains in some areas. Next slide. Each variant has 2 names, which adds a bit of confusion. The names point to the ancestry of the viruses and are recognizable because they contain periods. Next slide. Another source of confusion is the many mutations under discussion. Next slide. Focusing in here, the abbreviation E484K means that an amino acid called glutamic acid was replaced with a lysine at position 484 in the spike protein, which is a negative to positive change that can be problematic if occurring at a key binding site. This will be important later on. Next slide. This slide shows the U.S. COVID-19 cases that have been identified as being caused by variants of concern. B117 has been detected in 2,400 cases in 46 states, as shown at the top in the top map on the right. B1351 has been detected in 53 cases in 16 states, as shown in the middle, and P1 has been detected in 10 cases in 5 states, as shown at the bottom. Next slide. B117 was first detected in the U.S. during December 2020, but a study has shown that it likely arrived in November through multiple introductions. The current prevalence is estimated at 1 to 2%. Commercial diagnostic data suggests that we are in an early phase of logistic expansion, as shown in the graph on the right. 2 modeling studies suggest that B117 may predominate by late March. One of the studies suggests that high vaccine coverage will blunt the public health impact of the variant's higher transmissibility. Next slide. The receptor binding domain of the spike protein, shown as a trimer in the picture on the right, binds the host cell ACE2 receptor, an interaction necessary for infection. For SARS-CoV-2, the majority of neutralizing antibodies bind the receptor binding domain, blocking this interaction and neutralizing the effect of the virus. Convergent evolution of different variants has revealed a cluster of several mutations in the receptor binding domain, labeled and shown in yellow on the right, which increase the binding and infectivity and reduce the efficacy of antibody therapies. The mutations are N501Y, which occurs in all three variants, and E484K and K417T or N, which occur in the South Africa and Brazil variants. All of the variants of concern are distinguished by the large number of mutations that they have in their spike proteins. Next slide. There's also another variant called P2 that is the dominant strain in Brazil and has an E484K mutation with fewer spike mutations overall. Next slide. Next, I'll discuss the impact of these variants on vaccine effectiveness. Next slide. We reviewed 26 available studies of vaccines authorized or intending to be authorized in the U.S. and containing data on the ability of post-vaccination sera to neutralize SARS-CoV-2 variants. 8 studies were published and 18 were preprints. All had small sample sizes of less than 50 sera. 13 studies assessed only Pfizer vaccine, 3 assessed Moderna, 2 were on AstraZeneca, and 7 assessed more than one vaccine. 8 studies evaluated the effect of single or limited sets of mutations found in the variants and generally found minimal impact. Exceptions were E484K and the triple mutation in the receptor binding domain that I just highlighted, which had relatively large effects. The largest impacts observed were for the B1351, followed by P1 and P2, and then B117. In most studies, B1351 had a 3 to 11-fold reduction in neutralization, but ranged up to 97-fold. And B117 generally had a less than 3-fold reduction, but ranged up to 9-fold. A threshold of concern for reduction in neutralization activity hasn't yet been established. Next slide. In this dot plot, the reduction in neutralization activity of the vaccine sera is shown relative to the wild type or dominant strain used in each study, where no effect equals 1 at the bottom of the y-axis, and each dot represents one study result. Because no discernible differences were observed between the Pfizer and Moderna mRNA vaccines, and some studies presented combined results, the results for mRNA vaccines are pictured together in blue, while results for AstraZeneca are shown in green and Novavax in an orange or brown color. On average across the studies, B1351 had a larger reduction in neutralization than B117, and the 2 variants from Brazil, P1 and P2, had an intermediate reduction within the few studies available. Several studies noted that a single E484K mutation could account for most of the reduced neutralization of the South Africa and Brazil variants. One study assessed a B117 variant with an E484K change, which has been detected in the UK and other countries, and found that neutralization was substantively reduced compared to B117 alone. Next slide. 5 studies have shown that postponing the second mRNA dose may leave some people less protected against the SARS-CoV-2 variants. Notably, minimal or no neutralization against B1351 was observed after 1 mRNA vaccine dose. In a few studies, people who had recovered from COVID-19 and received 1 vaccine dose had moderate protection against B1351. All of the studies noted improved neutralization of B117 and B1351 after the second vaccine dose. 1 Pfizer vaccine study, shown on the right, noted delayed kinetics of the antibody response against the variants. Antibody responses to B117 and B1351 at week 2 after the first dose on the left were reduced relative to the dominant D614G strain, but improved slightly by week 3 on the right, especially for B117. Next slide. And both B117 and B1351 were improved at week 4, one week after the second dose. These results suggest maturation of the immune response could be important in protecting against the variants. Next slide. It is difficult to estimate how laboratory results might translate into clinical protection, given that there is currently no immunological correlative protection for SARS-CoV-2. In general, neutralizing antibodies in sera from mRNA vaccine recipients have been shown to be higher than for COVID-19 convalescent sera. The variation in the results I've presented may be explained by differences in experimental conditions, such as the type of neutralization assays used, differences in when and from whom the sera were collected, and whether recombinant viruses with limited or full sets of spike mutations were used versus viral clinical isolates. We also note that the antigen used in the AstraZeneca vaccine is not the pre-fusion stabilized spike protein used in other authorized vaccines, so results with this vaccine may have limited generalizability. Limitations for all studies were small sample sizes and a lack of generalizability. Many studies are preprints that are not yet peer-reviewed. Next slide. I'm switching now to data from clinical studies of vaccine efficacy or effectiveness related to the variants, and I'll move through the point estimates in this table from top to bottom. The Pfizer vaccine was shown to have high real-world vaccine effectiveness of 86% against symptomatic and asymptomatic disease in the UK and 94% against symptomatic disease in Israel during times when B117 was prevalent. Vaccine efficacy results for the Janssen vaccine clinical trial presented during this meeting were 74% in the US, 66% in Brazil, where 69% of cases were the P2 variant, and 52% in South Africa, where 95% of cases were B1351. Next slide. Importantly, the Janssen vaccine demonstrated similar VE against severe or critical disease across all 3 countries. In the Novavax clinical trial, a VE of 96% was observed for non-B117 strains, compared with 86% for B117 in the UK. Another trial demonstrated 60% VE in South Africa, where 93% of cases were B1351. For the AstraZeneca vaccine clinical trial, a VE of 84% was observed for non-B117 strains, compared with 75% for B117 in the UK. The trial in South Africa observed a preliminary VE of only 10% against B1351 and was halted based on this result. I'd like to caveat that this is a summary of the preliminary data that we have, and most of it has not yet been peer-reviewed. And this table isn't meant to compare across vaccines, as the studies used different outcomes and occurred in different epidemiologic settings. Generating additional data on the relative vaccine effectiveness for each variant would be useful. Next slide. In summary, the B117 variant is exponentially increasing in prevalence in the US, but has minimal impact on vaccine effectiveness. However, variants with additional substitutions in the receptor binding domain, like E484K, deserve special attention. The B1351 variant is currently at low prevalence in the US, but has moderate impact on vaccine effectiveness, suggesting that it would be prudent to start evaluating variant vaccines in case prevalence begins to substantially increase. The P1 variant has very low prevalence in the US, but has the same triple mutation in the receptor binding domain as B1351, which is worth watching closely. Additional data is still needed on the potential impact on vaccine effectiveness. Next slide. It is important to emphasize that the current prevention measures in licensed vaccines offer good protection against SARS-CoV-2 variants, and efforts are needed to increase the speed and degree of uptake. Periodic updating of SARS-CoV-2 vaccines will likely be needed. One modeling study has predicted that changing COVID-19 vaccine targets to target the faster spreading viral variants will be more effective than targeting the slower dominant strain, despite the initial prevalence of the variant. Next slide. Next, I'll discuss our response to the variants. Next slide. The SARS-CoV-2 Interagency Group, or SIG, was established by the Department of Health and Human Services to improve coordination among the agencies listed here. The SIG focuses on rapid characterization of emerging variants and monitors their potential impact on SARS-CoV-2 diagnostics, therapeutics, and vaccines. Next slide. CDC is undertaking a number of efforts to improve genomic surveillance and epidemiology that are relevant for monitoring the variants. As part of the National SARS-CoV-2 Strain Surveillance, or NS3, CDC processes approximately 3,000 random specimens per month from public health laboratories across the country. CDC has also partnered with commercial diagnostic labs to sequence an additional 6,000 specimens per week. CDC also contracts and partners with state and local health departments and universities to conduct genomic surveillance. In addition, CDC leads the SPHERES Consortium, which includes around 170 domestic partners and facilitates open sharing of SARS-CoV-2 sequencing data. Finally, CDC conducts focused molecular epidemiologic studies that are relevant to characterizing the variants. Next slide. This line chart shows the cumulative number of published SARS-CoV-2 sequences by collection date from laboratories across the U.S., starting in January 2020 to the present. The blue line represents U.S. sequences available in NCBI, the National Center for Biotechnology Information, and the orange line represents sequences available in GISAID, a global initiative that maintains a repository of viral sequence data. February totals are still pending. Next slide. Despite high vaccine efficacy, vaccine breakthrough cases are still expected to occur. Some will be caused by the variants, even if the vaccine has similar effectiveness against the variants. A vaccine breakthrough case is defined as a person with SARS-CoV-2 RNA or antigen detected in a respiratory specimen collected at least 14 days after completing a primary series of an FDA-authorized COVID-19 vaccine. Cases are identified from the National Case-Based Surveillance, the Vaccine Adverse Events Reporting System, as well as reports from health departments and healthcare providers. CDC works with state health departments on case investigations, and available specimens are sequenced to identify variants. Data from investigations will be posted or published when available. Next slide. Moderna and Pfizer have announced that they're launching booster studies of current vaccines in the U.S. and are also developing second-generation vaccines against B.1.351. Moderna's published strategy includes testing a vaccine specific to the South Africa variant in a multivalent vaccine that combines the previous vaccine for the ancestral strain and the new vaccine for the variant. Still yet to be defined are the framework of evidence indicating the need for a modified vaccine and the process for evaluating, deciding, and recommending whether a modified vaccine is needed. The WHO will likely have a role in global coordination and are developing a risk assessment framework. Next slide. Last week, the FDA published guidance on the evidence needed to support an EUA amendment for vaccines addressing the emerging SARS-CoV-2 variants. The required evidence includes information on good manufacturing practices and controls, non-clinical data from laboratory studies and animal models, clinical data from immunogenicity studies designed as non-inferiority comparisons with the previously licensed vaccine to evaluate either a primary series or a booster dose. Studies could be from a single age group with extrapolation to other age groups, and safety data could include those generated during the immunogenicity evaluation period. Information on laboratory assays and immunogenicity endpoints is also needed. Next slide. In closing, we will continue to monitor evidence on the emergence and spread of SARS-CoV-2 variants, vaccine effectiveness, breakthrough infections in vaccinated or previously infected persons, and the ability of post-vaccination sera to neutralize emerging variant viruses. The work group in ACIP will review evidence submitted for booster doses and any next-generation vaccines if it becomes clear that they're needed to address the variants. Next slide. Thank you for your attention.
Thank you very much for that very interesting and informative presentation. I didn't find that in my packet of material sent to me, but it's possible that it has been sent afterwards. Can we make sure that members of the voting group have it, please? Thank you. It's open for questions and comments at this point. Let me begin with one. Could you tell us what is known about recombination and whether recombination at all has played a role in development of these transmission of these newer mutations or among the viruses?
Well, I know that SARS-CoV-2 does not have a segmented genome, so unlike influenza, where this is a major source of it generates a lot of different variation that way. I've seen limited reports that it's possible for SARS-CoV-2, but I don't think it's not clear that it's a major source of evolutionary variation.
Yes.
I wonder if anybody from the lab.
Actually, I'm sorry, I messed up my terms. I'm sorry.
Sorry.
I'm not speaking so much of recombination, but for poliovirus, for example, the RNA will slip down, jump off, and jump onto another virus that's in the same area. Okay. And I'm blocking out a term. Forgive me. I used the wrong term. Showing my ignorance.
I've seen a few papers that mention that may be the case, but it doesn't seem to be a major source of introduction of mutations. But does anyone from the lab want to comment further, Natalie or Dave?
This is Dave Wentworth. I think it's a good question, and we do see in coronaviruses RNA recombination, like you mentioned, and but we haven't seen it drive the evolution of these variants. These variants have independently evolved and look, in a linear sense across the genome as point mutations that have occurred and then develop further point mutations. Of course, when so many mutations are happening at the same time, like in the 351 variant that Heather discussed, you can't rule out recombination. We just don't have evidence of it because that would be intra-spike recombination events. Usually with coronaviruses, you'll see the recombination being, everything downstream, everything 3' to some point, and it's easy to see. Over.
Thank you very much.
Can I make a comment? This is Bonnie Maldonado. So recombination events are actually quite frequent with RNA viruses. For example, with polio viruses, they occur on a regular basis, and they're usually not canonical, so they can happen at any point. And there are recent non-peer-reviewed papers, actually from Mark Dennison's group, suggesting that there are recombinations that can occur. Again, these are different from these specific canonical mutations, but it's not insignificant.
Thank you. Dr. Zahn.
Thank you, Dr. Romero, and thank you all for this great presentation. I just make a comment that on a local public health level, it would be great, and I know this is all in development, but it'd be great to have a roadmap or a sense for how a variant becomes a variant of concern. We've had the experience in California, and I think this is going to happen again and again, from researchers will identify new strains out there, and they may meet both the medical literature and the popular media, and there may be speculation as to how important new strains are and whether or not they're more infectious and whether they change the epidemiology. And when we get asked about it at the local public health or state public health level, it would be great to have a roadmap or a sense of how, when these are identified, further investigation develops. And I know this is developing. I know you talked about it a bit in your presentation, but the more we can have a way to describe how we're assessing these events, it would certainly make it easier in terms of public communication. Thank you.
Thank you.
Adam McNeil, do you want to take this one? Are you there?
Sure. Hi, good afternoon. This is Adam McNeil. So I'm with the Epidemiology Task Force. Just to mention, this is a very active area of discussion. From the U.S. government standpoint, we have been working on developing definitions which are in the interagency space. I think we're also looking at internally having processes for trying to, basically make committee decisions in terms of what constitutes a variant of concern. I think it's become very important that we need these clear definitions. WHO is also working on trying to come up with a definition. Maybe just to briefly mention some of the key criteria, at least, that go into this include evidence of the virus having immune escape, as Heather spoke quite a bit about, evidence of convergent evolution, potential impact on diagnostics or therapeutics, or evidence of increased transmissibility or disease severity.
Thank you. Dr. Goldman.
Thank you. So I noticed Florida seems to have a disproportionate number of variants. Do we know why it is so concentrated in this state? Is it a aspect of testing and or not looking for it in other states, or is there something about the population that we seem to have more variants here?
Adam or Dave, would you like to answer that?
Sure, this is Adam. I can jump in, and if Dave wants to add more. There's a couple things, particularly with B117. Early on, there was using what was called the S gene target failure. Basically, there was a number of different diagnostic PCR assays that were indicative of the deletion of amino acids 69 and 70. So early on, using this S gene target failure, basically as a screening assay, there were attempts to rapidly try to identify circulation of B117, and this somewhat depended on which diagnostic lab was involved. So I think there is a natural concentration in terms of certain states and enrichment of B117. That said, there probably is, in addition to maybe some of the skewing of the data, I think we're seeing more and more transmission, particularly in Florida. So it may be partly artifactual, but I think there is evidence of the virus continuing to become more prevalent. I think the other related thing is, given the evidence that B117 has a higher reproductive number, once you start hitting a critical point, obviously growth of that variant as a variant share is exponential. So it may just be partly a founder effect where a state like Florida had earlier introductions, and thus it started the exponential growth phase quicker than some of the other states.
Thank you. Dr. Maldonado. Dr. Kimberlin.
David Kimberlin, AAP Red Book. On slide 20, you mentioned that the AstraZeneca vaccine being studied at the time in South Africa was not a pre-fusion stabilized spike protein, and therefore it limited generalizability to other vaccines. Can you remind me, for the two mRNA vaccines and for the Johnson & Johnson Janssen vaccine, are they pre-fusion stabilized spike proteins?
Yes, they are.
So AstraZeneca was unique in not being a pre-fusion stabilized spike protein among the three that are authorized currently.
That's my understanding.
All right, thank you.
Dr. Cotton.
Thank you for mentioning immunocompromised host as an area of emerging interest. As someone who cares for many immunocompromised hosts, we are seeing an increased rate of refractory disease with months of infection and disease. And I'm really concerned that this may serve as an important reservoir for multiple mutations, and then there could be nosocomial transmission, et cetera. So I guess I'm wondering, will this group be an area of focus either for vaccinations and or for research? As I'm worried that this may be the most significant problem population.
Dave, do you want to talk about anything you're aware of from the lab side in this area? Or Adam, I don't know if you're aware of any epi studies.
Yeah, this is Dave Wentworth. I'll start off, but we don't have particular lab studies geared towards this at the moment. I do agree that this is an important population, and there are specific studies periodically where they're not studies, but they're specific cases where people refer to the CDC, about a long-term shedder that it can't resolve the infection, et cetera. But we don't have a study that I know of from the laboratory side, and there may be some in the epidemiology division. Thank you.
Yeah, so this is Adam. I'm not aware of any studies. We can follow up further, but I'm not aware of any studies specifically looking at immunocompromised individuals. I think this is a very important point, and I know that was one of the prevailing hypotheses behind the emergence of B1351 and the fact that there were, quite a number of mutations that emerged within a single variant is that it is possible an immunocompromised individual, in essence, shed for a long period of time, which allowed the virus to acquire quite a number of mutations. But I think this is an important point, and, there may be opportunities thinking about locations where there are high prevalence of HIV positive individuals, as one can think about the importance of potentially doing enhanced or further surveillance. A very good question.
Thanks. It's, for the record, just immunocompromised in general, and I would say actually not even as much HIV as much as patients on rituximab and other really potent immunosuppression. And we are not currently referring these cases to the CDC, so if people wanted that to be done on a large scale, I think the immunocompromised host clinicians would need to know about that so that we could track that down.
Thank you.
Thank you. Dr. Sanchez.
Thank you, Dr. Romero. I had a question about the emergence of the UK variant, and was it at all related? There's been a lot said about UK delaying the second dose, and has there been any studies looking at that in terms of the relationship? And then second of all, I think your presentation was just great and very enlightening. Really answers to me the question that was posed by the first speaker, the first talk this morning, about whether we should continue with the 2-dose regimen. And I think this clearly answers that we need to go with our current regimen, at least with the messenger RNA vaccines, that they should be given us a 2-dose schedule and not delay the second one. So it kind of just puts, in my mind, to rest that first question. But I was wondering whether there was any data looking at delays of second dose with any emergence of these variants. Thank you.
Well, I just wanted to be clear that sequentially B117 did arise and become prevalent before the delay in the second dose became a policy. I'm not aware of studies looking at the delay of the second dose. There are quite a few studies being published in the literature now on real-world vaccine efficacy of 1 dose, and I believe those were used as a rationale to support the policy in the UK. But I think it's a good question, and the more conservative approach is just to go with what we have right now in terms of the two doses. Thanks.
Dr. Long.
Yes, I'm wondering if there is any signal that children are affected differently with the mutant variants.
Adam, could you take this one?
First of all, maybe just to mention the overall prevailing thought outside of B117 is that these variants in general, so not specific to children, don't have differing impact on clinical outcome. There is some data, it has not been actually published yet. I don't think there's even anything available in pre-publication, just a report and review of a number of studies out of the UK that indicate that B117 may be more associated with increased severity, both in terms of hospitalizations as well as death. But that said, I'm not aware of any evidence as of right now specific to children and differences in terms of outcomes.
Us pediatricians we pediatricians are concerned about the next respiratory season. We don't really know from whence comes the blessing that children seem to be less affected by the SARS coronavirus 2, but we're all kind of concerned that if part of that explanation is some cross protection or being primed with other coronaviruses, that we will have a very complicated next winter when we just haven't had any respiratory viruses this winter. So we'll have a double group for their first influenza, the double group for the first RSV, and potentially children who have not seen coronavirus. So that I think it will be important, and we should be careful to be sure to understand the genetics of the organisms that have caused disease in children. Thank you.
Thank you. Dr. Gluckman.
Thank you. Very interesting presentation. My question has to do, as you've noted, that both Moderna and Pfizer are launching booster studies for and developing a second-generation vaccine. One is, do we have any idea what the speed to market might be for a second-generation vaccine? And secondly, do we believe it's going to be consistent? It's going to be important to have consistency in the platform to whatever the patient may have received on the first vaccine. So in other words, if they had their primary vaccination with an mRNA vaccine, any second generation should be with mRNA vaccine. If they had their primary immunization with the Janssen 8026 platform, that the second generation vaccine should be on the same platform.
Thanks. This is Dr. Oliver. I think we're going to have to wait and see both with kind of speed of how this goes, as well as what the recommendations will be. Like was mentioned before, we know FDA has kind of started laying the groundwork for how this would work. And so yet again, we would, wait for data to be submitted to FDA. We would, take a look at it and then bring it to ACIP for future discussion and recommendations. As been publicly stated, I think Moderna has said that they're starting trials soon. So we'll keep an eye on that with the variant vaccines, and we'll bring any updates that we have to ACIP on these vaccines moving forward. Thank you.
Dr. Dushan.
Thank you, and thank you for the presentations on this critical work. It seems with the predicted prevalence of B117 in the U.S. and the increasing recognition of other variants that we remain vulnerable to a potentially severe fourth wave of this COVID-19 pandemic. And it's clear that vaccines ultimately will be our best weapon against COVID-19 and its variants. But I think it's important in this discussion to recognize that at the moment it can't be our only strategy, and that we really need to reinforce the importance of our other COVID-19 prevention measures at this time, including limiting activities and well-fit and well-made face masks, and paying careful attention to ventilation and avoiding crowded indoor spaces. I noticed that one slide mentions that high vaccine coverage will blunt the impact of higher transmissibility, and clearly the more people that are vaccinated, the more are protected. I'm wondering if you can tell me more about what high vaccine coverage means in this context. Thank you.
Thanks. I mean, I think your points are well taken. That particular study was published in the MMWR. I don't have the details in front of me. I don't know, Adam, I know you had presented on this recently. Do you remember what the assumption was for that study?
Yeah, I would have to go back and look. Our modeling team set up dynamic models for this. I think they had modeled it based on vaccine coverage increasing over time, but I'm not immediately sure of the exact assumptions that went into the model.
Okay, thank you.
Heather stated it's published in MMWR.
Thank you. Dr. Weiser.
Hi, thank you for such a fascinating presentation. My question has to do with the adequacy or the completeness of the surveillance system, and particularly for rural areas where most of our facilities with IHS are located. And I'm just wondering if we are getting adequate representation from those rural areas, or are there pockets that might be being missed? And then related to that, if we're interested in trying to participate in this, should we be reaching out to our state health departments or to CDC? Thank you.
Adam, do you want to take this as well?
Sure. So representativeness is certainly a challenge, particularly for a large proportion of the pandemic. We were relying primarily on, platforms that involved crowdsourcing of sequencing. So recently, as Heather mentioned, there are a couple different initiatives that we've taken on to really try to scale up the number of sequences as well as the representativeness of these. This includes the NS3, so the National SARS-CoV-2 strain surveillance, which functions through our state and jurisdictional public health labs, as well as trying to, across the board, generate sequences from a large number of specimens from commercial laboratories. So I think we're getting much closer to being representative. We're almost there where we have a denominator that we're confident in, so we can start actually generating representative prevalence estimates. So that should be coming out soon. In terms of participation for NS3, it is functioning through state and jurisdictional public health labs. So working with them would be the way to operate through NS3. But just to underscore, there are a number of other sequencing efforts through state and local public health labs as well as academic partners. So I think as much data as we can get in the public space is certainly going to be helpful and contribute to the overall effort in terms of understanding viruses that are circulating.
Thank you. Dr. Dushan?
Sorry, I'm really bad. I'm taking my hand down. My apologies.
I'm just making sure that nobody gets left out. Dr. Arthur?
Hi, Phyllis Arthur from the Biotechnology Innovation Organization. In response to a question raised earlier about the different data, it might also be good for the ACIP to take a look at emerging data on the use of the monoclonal antibodies in the preventive space. That could add some complexity to the discussion about prevention particularly in the presence of the variants, but some of the preliminary data is quite strong, and it might be worthwhile having some opportunity for the ACIP working group and members to look at that data as it starts to roll out.
Thank you. Are there any comments, any other comments or questions? I'm not seeing any. Dr. Maldonado.
Yeah, so, sorry, just one other comment. I think in light of the data around the variants and some of the many models, including the CDC's models that have been circulating recently around the impact of these variants, potentially with large ranges, of course, on transmission, therefore modifying our NOTs or RTs and impact on herd immunity and percent vaccinated. I wonder if at an upcoming discussion we could see more of a presentation around that, because I do think that there are a few models out, several models now out there, including the CDC, and it might be helpful for us to see how that impacts our concept of herd immunity. And those are suggesting that whatever you define herd immunity as, and how many people you think are already infected in the U.S., those can be reached anywhere between May of this year and May of next year, or maybe beyond, depending on the variant. So I think it would be helpful for us to see a summary of some of those models if we have time at some point in the future.
Thank you. Any other comments or suggestions? I think that's it. So I'll turn it back over to Dr. Cohen to see if she has any final words.
I don't. This concludes our meeting. Once again, I just want to thank all of the incredible ACIP members, ex officios, and liaisons for sticking through with all of us for 4 out of the last, I think, 6 days. I think that this, since it is March 1st, we're going to count this as our March meeting, and hopefully we will not have to meet again in March. But we will respond if needed, and we anticipate another emergency meeting before our next scheduled June meeting, and we may also be having a half-day regular meeting, which would not occur until May, where we'd have enough time to announce, to go back over some of our non-COVID issues that we didn't have time to discuss last week. Dr. Romero, I think we're ready to close the meeting.
Again, thank you all for your time and your dedication. I'll gavel us out with my knuckles. This session and meeting is closed.